Hey gang, we've got section 8.5 here talking about regulating enzymes, specifically allosteric regulation. But the first concept here is in a cell, you can't have all of your enzymes all working all of the time. And a cell would have options on how to control when certain reactions are happening and when they aren't. A bad example or a bad way of controlling this would be to say, hey, I need a lot of reaction A. So let's make a ton of the enzymes to cause reaction A. And then later on, when you don't want as much reaction A, if the cell just destroyed all of those enzymes, that'd be a really bad move because it would waste a ton of energy. It takes a lot of energy to make an enzyme. So that would be a bad option. Although it is an option, you can destroy enzymes and create enzymes as you want reactions to happen. A better option would be to turn enzymes on and turn the enzymes off. And when you want, if you've got a whole bunch of enzymes in a particular cell and you want them to do more work, then you can activate them. And if you want them to do less work, you can inhibit them, causing them not to do their work. And then you're not creating and destroying all the time. So that's kind of the idea there with this regulation. And then allosteric regulation specifically is regulation that happens regulation, some sort of molecule regulates a enzyme at a spot that's not the active spot. So it would be like a non-competitive inhibition if it was inhibiting it. And there's also activators that do a similar thing. But first idea is on this side of the diagram here. It's showing that most enzymes, this happens a lot, but or I should say many enzymes, are going to be linked together. So we've actually got 4 different enzymes here. 1, 2, 3, 4 different enzymes that are connected together, and they're going to influence each other a little bit. So that's the one idea. They call that an enzyme complex, and so that's a very common thing for there to be enzyme complexes. The other thing that's very common is that enzymes aren't just these things that sit there waiting for a substrate to come to them. They actually have a certain amount of activity that they do on their own. And that activity is that they will change shapes, and so there will be like an active shape, and then it will oscillate, they call it, but it'll just flip back and forth between an inactive shape and back to an active shape. Okay, and this happens very naturally, going back and forth between these 2 different shapes, back and forth and back and forth and back and forth. So if you want a reaction to happen more often, one way to do that would be to just kind of lock it in the active state. If it's in the active state, then all those active sites are actually functional. And so allosteric regulation says you can take a molecule, an activator, jam it in there, if you will, and it's going to keep all of those at in an active form or an active state, and then that's going to increase the chances of a reaction happening. Okay, so that would be an activator, an allosteric activator. An allosteric inhibitor then is something that would fit into that same regulatory region. It's not the active site, and lock it into the inactive form. And so if we wanted less of a reaction to happen, we could lock it into the inactive form. Okay, an example of where this actually happens, if we look up here at the I got a little picture of glucose here. So here's glucose. We're going to study a bunch about this in a couple chapters, but the whole process of turning glucose into carbon dioxide through cellular respiration, obviously it's also going to produce water and it's going to require oxygen and things like that. But in general, if we're going from glucose to carbon dioxide, it is a pretty crazy number of steps, like something like 30 steps. 30 different reactions, which means 30 different enzymes. It might be even more than that. I'm just throwing a number out there. But there's 30 steps to this. And the whole reason that your body is doing the glucose to carbon dioxide thing is to make ATP. And I should have an arrow here, but the goal is to make ATP. So this glucose to carbon dioxide, that's going to be releasing energy, so exergonic. And that exergonic reaction, the energy that's released from that is going to go into producing ATP. Okay, which is what your body uses to store energy a lot of time in the cell. Anyway, this 30 steps of reactions, if it is not happening very much, then your cell would actually get very low in ATP. So there would not be very much ATP over here, but there would be a whole lot of ADP. If you remember, ATP and ADP, there's that cycle where ATP, as it's used, cycles back to ADP. Okay, so if we've used up all the ATP, there'd be a lot of ADP, and we're running short on ATP, so we would want our steps of reactions to happen more often. Okay, so we want all these steps of reactions. So ADP ends up being an activator in step number 6 or something like that, but one of the early steps. ADP is actually an allosteric activator on one of the enzymes back here somewhere, forcing or causing this reaction to happen more and more if there's a lot of ADP. Also, ATP, if we've done a whole lot of sugar breakdown and we haven't used up much ATP, we'd end up having a large quantity of ATP in a cell. That ends up being an allosteric inhibitor. And so if there's lots of ATP produced, it'll actually fit into that same area, the enzymes in that same one of those early stages, and it will actually inhibit that, which means the entire process will shut down. Boom, no more. And it'll do that until we run out of ATP, and then we have a lot of ADP, and that will activate it. Okay, and so that's an example of when this happens, this allosteric regulation idea as a way of making sure that enzymes work more or work less depending on what the cell actually needs. The next idea here is something called cooperativity, and when we're looking at these enzyme complexes here, like I said, they're joined together and they influence each other, and so oftentimes, if one of them is actively working on a substrate, so there's a substrate in the active site, that means that one obviously has to be in the active state. This cooperativity idea is that if that one's in an active state, not because of an inhibitor or an activator or anything like that, but just because of the substrate, it'll actually lock the other ones in that complex. It'll lock them into an active form as well, which increases the chances that if there's one substrate molecule around, there's probably more. So if I've locked these into the open state, it means that as this one's working, it's increasing the chances of these other ones working as well. And so we get this idea of them cooperating together or working cooperatively, and so that is that idea. Next we're going to talk about feedback inhibition. Okay, the concept of feedback inhibition, I kind of went through with the whole ATP idea before, and the fact that ATP will inhibit a reaction that happens very early in the process of making ATP, but kind of feeds back, one of the products feeds back to the beginning. But here's another diagram here where we start out with an initial molecule, initial substrate, and it is acted on by enzyme 1, okay, and then that makes a product which is acted on by enzyme 2, which makes a product which is acted on by enzyme 3, and so on. And so these are continuing to be worked on by a whole series of enzymes. That series of enzymes makes some final product. Isoleucine here is what they're talking about. But anyway, that end product ends up going back to the initial enzyme, one that's way back at the beginning, and being an inhibitor of that first enzyme, stopping the entire thing here. So if I have produced a lot of product, then it will end up increasing the chances that this entire system stops working. Okay, and so that's feedback inhibition. Oftentimes, just to kind of throw this out there, we've got these like 5 enzymes here that are all working together to do something in the end. A lot of times these enzymes will actually be held together in an enzyme complex, and they're different enzymes, but they'll actually be held together to increase the chances that the products from enzyme 1 will actually make it to enzyme 2 versus if they were in total different parts of a cell. Another thing that will often happen is these enzymes will be locked into a membrane sometimes. So they might not be connected with each other, but they'll actually be locked into a membrane somewhere, so that on a membrane enzyme 1 is close to enzyme 2, close to enzyme 3, and so on, again increasing the chances that the products will go down the pathway. So otherwise you'd have enzymes just kind of floating around the cell, and you'd have to just wait until hopefully one of the product A here, intermediate A, finally finds enzyme 2 wherever it happens to be floating around in the cell. So that's it. We're done with chapter 8, and hope that you are enjoying it, the wonders of molecular biology, and we'll see you in class.